All right, well, it looks like the majority of people have been able to enter this morning. Well, welcome to the Visual Sonics live webinar series. My name is Sarah Burris. I'm the scientific liaison here at Fujifilm Visual Sonics. And we're very excited to have our presenter, Rowan Swann, today talk about her work about the development and evaluation of a bone-targeted photoacoustic imaging agent. Now, before we get started with the webinar, we just have a few notes regarding today's webinar. Just as a reminder, we are recording this and all lines are muted, so a recording will be made available at the end of the day today. If you have a question, please submit that through the Q&A button located at the bottom of your screen. And questions will be answered at the end of the session. And you can expect the webinar to last somewhere between 25 and 30 minutes with another 5 to 10 minutes for questions and answers. Our speaker today, Rowan, received her bachelor's degree in pharmaceutical sciences at the University of Ottawa, where she had the opportunity to gain research experience in government and academic laboratories. Throughout her time in Ottawa, she worked under the supervision of Health Canada scientist, Dr. Stephen Brooks, and University of Ottawa researchers, Dr. Tony Durst, and the Vice Dean of Science, Dr. Louis Berriot. Upon graduating in 2018, she began studying at McMaster University to fulfill a master's degree in chemical biology. And in 2020, under the supervision of John Valiant and Dr. Sam Sadeghi, Rowan transferred to the Ph.D. program. Her research endeavors have focused on evaluating various molecular targeted photoacoustic agents using small molecular near-infrared dyes, such as IR-783 and methylene blue, as well as carbon nanotube-based agents. Use of antibodies for cancer-targeted imaging and small molecules for bone and infection-targeting imaging are the current focus of her work. So with that, I give you Rowan.
Thanks. All right, let me just get my slides here. All right, I trust everyone can see my slides and hear me. So thank you all for joining today. I'm really excited to tell you a little bit about the work I've been doing towards bone photoacoustic imaging. So I thought I would start just with a pretty general research overview. Up to this point, my research has focused on designing molecularly targeted photoacoustic probes using organic reactions, such as the one I'm showing here, which is the inverse electron demand Diels-Alder reaction that occurs between transcyclooctane and tetrazine. So this reaction is a bioorthogonal click reaction with extremely fast kinetics, which allows us to use in vivo pretargeting strategies where essentially we administer a targeting vector and a diagnostic agent separately with the intention of increasing target to background signal as well as reducing toxic accumulations in healthy tissue. Another aspect of this reaction pair that we can make use of is development of modular imaging and diagnostic probes. For example, we can interchange diagnostic agents for therapeutics, as well as interchange the targeting vector that we're using. So the overall goal of my research has been to utilize organic reactions such as the inverse electron demand Diels-Alder to facilitate the connection of photoacoustic dyes with various targeting vectors, and to then validate their utility in vitro and in vivo analysis. So as you probably know by now, the focus of this work is bone targeting. And to do this, we are utilizing a class of molecules called bisphosphonates, which have been widely used in both the literature as well as clinical settings to target bone and pathological calcifications, which is due to their affinity for hydroxyapatite, which is a calcium-containing mineral that's present on bone during remodeling processes which regulate blood mineral concentrations, as well as to accommodate bone growth and degradation. So one thing to note about bisphosphonates and hydroxyapatite is that we can target healthy tissue as well as disease states, since there's always a level of hydroxyapatite present on healthy bone. But it's important to note that in disease states, especially where there's bone degradation happening, hydroxyapatite presents in elevated levels, which could act as a sink for these bisphosphonate-derived probes to accumulate at. So although we should see binding to healthy bone, we would expect elevated signal at disease states. So now you might be wondering why we want to image bone with photoacoustic. Well, optical imaging does have the ability to address a few of the existing limitations associated with conventional X-ray techniques. Namely, we are able to have reduced exposure to ionizing radiation, which is something of a concern for younger or immunocompromised patients. As well, we have the ability to provide functional information related to the tissue of interest as well as the surrounding area through monitoring of endogenous and exogenous chromophores. In terms of optical imaging, we might choose PA over fluorescence because of the improved depth resolution we can gain with photoacoustic imaging, which is a result of the reduced acoustic attenuation that occurs in tissue compared to light. As well, the current state of photoacoustic bone imaging has been limited to the use of endogenous contrast alone or passive targeting strategies, which allows us to warrant having a molecularly targeted photoacoustic probe, which is exactly what we've worked on here. So the design of this probe is basically split up into two sections. We have our TCO derived bisphosphonate or compound 2, which was previously synthesized in our group and evaluated for its ability to target bone, as well as our IR783 tetrazine or compound 5, which is a novel tetrazine derived dye that we synthesized in 2 steps using the commercial fluorophore IR783. So throughout the presentation, you might see a few of these compound numbers floating around. Just be aware that when I indicate compound 7, that's indicating that we have combined the TCO bisphosphonate and the IR783 tetrazine to form the targeted probe. And I denote an AT or a PT to indicate whether we've chosen an active targeting or a pre-targeting approach in the study of interest. So overall, throughout this work, we're using a healthy mirroring model to demonstrate the ability of the photoacoustic probe to bind and detect regions of active bone turnover using 3 different delivery strategies, which I've outlined in this figure here. So the first is a local injection where we inject the actively targeted probe directly into the bone and joint space. And then we also have 2 different systemic injection approaches where we either inject the actively targeted probe or we use a pre-targeting strategy where we first inject the targeting vector, and then after a certain amount of time has passed, we can inject our photoacoustic probe and allow the reaction to occur in vivo. So just to highlight a few of the imaging specifications that we've used throughout this study, we utilize the Vivo Laser X system equipped with the MX400 ultrasound transducer. And anytime I'm showing in vitro photoacoustic analyses, know that these were performed in the contrast agent phantom tubing and the material was stationary within the tube. So the first thing we set out to do was characterize the photoacoustic signal of our compounds in whole murine blood. So in figure A here, I've just shown the overlaid photoacoustic spectra of each chromophore of interest. So we have oxygenated and deoxygenated hemoglobin, as well as compound 7 and compound 5. One important thing to note about this overlay is that you can kind of see that there's a bit of overlap between the signal of our dyes of interest and deoxyhemoglobin, which I can tell you right now definitely presented a few obstacles to us throughout our studies. But one of the main things we were able to show in this data set here is that we should still be able to detect our dye even in the presence of deoxygenated hemoglobin. And we were able to show that presuming there's at least 20 micromolar of our dye present in a specific location, we should be able to distinguish it above background signal of our oxy and deoxyhemoglobin. With that, we moved to an in vitro binding study where we wanted to assess the specificity of our dye targeted to hydroxyapatite using using both active and pre-targeting strategies. So to detail, we incubated our targeted or non-targeted dye, either in the presence or absence of hydroxyapatite. And after centrifuging the samples, we analyzed the supernatant via absorbance to determine how much of the dye had bound to the hydroxyapatite using the calculation here. So in figure A, I've just shown the percent binding values that we acquired from this work, where we were able to show the specificity of the bisphosphonate for hydroxyapatite using both active and pre-targeting approaches compared to our non-targeted dye, which experienced close to no binding. We took this study a step further by analyzing the hydroxyapatite pellet via photoacoustic imaging. So essentially what we did there was we washed the pellet and then we injected it into the phantom tubing and analyzed the signal that we were able to acquire. So from this data, we could see that compared to both vehicle as well as our non-targeted dye, both active and pre-targeting strategies showed elevated photoacoustic signal present on the hydroxyapatite. And you can also see that visually in figure C here, which is exactly what we were hoping to see. So with that, we decided to move to a few in vivo models. But before I jump into that data, I just wanted to give you a bit of an idea of how we are representing our data here. So in each of the studies, we use 3D multi-wavelength unmixing scans, where we're unmixing the signal from our dye with oxy and deoxyhemoglobin. Just to note, I do subtract the signal from oxy and deoxyhemoglobin from each of the images. So the green signal that you're seeing is going to either be some sort of background signal that the machine picks up or the dye itself. So to study the ability of this compound to bind bone, we were looking at the hind limb of each mouse. So our scans took place over across the hind limb. And I'm just showing here the way we chose to represent the data through texture mapping or render modes, and highlighting where you can see the femur and the knee joint in each of the images. So that's the femur here and the knee joint in the render. So although ultrasound hasn't really been popular for bone imaging, especially because it is not well equipped for the tissue density differences that you get between bone and the other surrounding tissues. We're actually using that to our advantage here because the bone casts an acoustic shadow underneath of where it's located. So when we go through our two-dimensional sections that make up our three-dimensional images, and I'll show you in this Spectro scan right here, we see this acoustic shadow anywhere that bone is present. So we were able to use the 2D scans to define regions of interest that were really tight around the bone and knee joint for our 3D signal quantifications. So in addition to these multi-wavelength unmixing scans, we also performed Spectro scans, where we were able to acquire a spectral curve of the signal arising from a specific region of interest. And essentially we aimed to show that the signal that we were seeing wasn't just some sort of background signal that the machine might have picked up. It was in fact due to our dye. So here I'm just showing you what a typical background signal does look like. We can see the main peak of deoxyhemoglobin and potentially some oxyhemoglobin later on in the spectra here. So the first photoacoustic imaging study was designed to assess whether our actively targeted probe was capable of binding bone when delivered directly to the femur and joint. So in this study, we were performing intrafemoral injections of either the control dye or non-targeted versus the actively targeted probe here. We imaged the mice pre-injection as well as 4, 24, and 48 hours post-injection. And finally, we took ex vivo scans, basically as a way to verify that the signal that we were seeing in vivo was attributed to signal within the bone as opposed to the surrounding tissue. So before I show you that data, I just wanted to give you a little idea of how we set up these intrafemoral injections. They were quite complicated and tricky to perform. So essentially, if the mouse was laying on its back and the femur was positioned approximately 90 degrees to the countertop, we were able to get a direct pathway to the femur with our needle. And we used this ligament that you can actually see through the skin to line it up so that it would hopefully inject the dye right in the head of the femur here. Unfortunately, the injections were a bit inconsistent, but we were able to come up with a list of criteria that we wanted to meet throughout these injection processes in order to decide whether the mouse was going to give us accurate results or not. So we did use a few extra mice than what we normally would have. And this allowed us to exclude a few of the mice from the study that we could tell the injections did not go as planned. So here is the data from that study. Just in figure A here, I've shown the 3D multi-wavelength unmixing scans in render mode. And I've shown the control versus the targeted groups at each of the time points and ex vivo. So from this data, we were able to visually interpret that the targeted dye appeared to be retained in the bone over the course of the study. Meanwhile, the non-targeted control dye appeared to wash out rather quickly. And we were able to verify this using both our 2D spectral analysis as well as our signal quantification. So in figure B here, just shown the spectro scans where we define a region of interest around the bone, and then we're able to acquire the spectral curves from within those regions of interest and compare them. So that's what I'm showing here. And from this, we were able to see the very characteristic double peak system that our dye usually presents. And that was present in the targeted group. Meanwhile, in the control group, we did not see very much in the way of variation between that and the background signal. So this was showing us that the signal in the targeted group that we did have was in fact due to the presence of our dye. So after quantifying the signal in 3D, we were able to show that we achieved statistically significant signal in our targeted group out to 24 hours post-injection that was above both the background signal as well as our non-targeted control group. So from this data, we were able to demonstrate that our targeted dye was retained within the bone out to 24 hours and washout of our non-targeted dye occurred pretty rapidly. With this, we moved to an in vivo active targeting study where we wanted to assess whether the actively targeted probe would be able to reach sites of bone turnover upon systemic injection. So for this study, mice were either injected with the control dye or the actively targeted dye via the tail vein. And mice were imaged pre-injection as well as 4 hours post-injection and again ex vivo. So as you can see in figure A here, I've shown the 3D multi-wavelength and mixing scans in texture mapping mode. The top row is our control group and the bottom is our targeted, and we have each of our imaging time points displayed. So a few things I want you to focus on in this data. First of all, you can see exactly where the femur is, and comparing our 4 hours post-injection with our pre-injection, we were able to see quite a bit of a visual, quite a bit of contrast available at the site of our bone 4 hours post-injection in our targeted group. And when looking at that signal ex vivo, we could see that it was definitely present within the bone and not just within the surrounding tissue. Compared to our control dye, we had quite a bit more signal available in the targeted group, although there was a few speckles of signal throughout our control group that were presented as well. So the 2D spectral analysis from these regions of interest that I'm showing here, we were able to show that our targeted dye was definitely present within the bone at 4 hours post-injection. And in our control group, again, we didn't see a huge difference between the control and the background pre-injection sample. So this indicated that there really wasn't much of the dye present in the control group. However, when we went to quantify this signal, we did see some non-negligible signal in our control group above background. And this we attributed to the fact that there could have been some of our control dye floating around in circulation still. So we can see that it's not actually at the bone, and we did our best to define the regions of interest appropriately. However, there was some signal within the control set of animals. But when you compare that to the data that we acquired with the targeted group, we could see that the targeted dye had far greater signal above both background as well as the control, which indicated that the presence of the bisphosphonate was able to localize greater quantities of our dye to bone. So overall, this data demonstrated the specific uptake of our targeted dye to bone using our pre-clicked or actively targeted dye. So this brings me to our final study where we wanted to assess a pre-targeting strategy and how it could potentially deliver our dye to bone. So in this study, we split the animals into 2 groups, pre-targeted and control. In the pre-targeted group, the first injection was of our TCO bisphosphonate, or compound 2, and our control group were injected with saline. After 1 hour, we then injected all of the mice with IR783 tetrazine and waited 4 hours. So the mice were again imaged pre-injection as well as 4 hours post-injection of compound 5. And again, ex vivo to verify what we see in vivo. So as you can see in the multi-wavelength unmixing scans, we did acquire some signal within the bone of our pre-targeted group, especially compared to the background signal. However, in our control group, we definitely did see a little bit of signal in vivo, and ex vivo, it seemed that we could for sure see some signal within the bone. Although compared to the pre-targeted group, it seemed to be minimal. After analyzing the spectral curves of the bone within the bone of each of the mice, we could see that indeed in our pre-targeted group, we had our dye present. And even in our control group, it appeared that there was a few differences between that and the background or pre-injection image. So we were able to determine that, yes, there definitely was a little bit of our dye present in the control group as well. This was also evident when we quantified our photoacoustic signal. There was elevated signal in our control group above background. And again, we attributed this to the compound 5 remaining in circulation throughout the study. which was logical to us because we actually did increase the concentration of 5 that we injected as a way to hopefully promote the in vivo click reaction to happen, although we probably could have done without that after seeing the results of the study, because in our pre-targeted group, we certainly did have elevated signals at 4 hours post-injection compared to both background as well as the control group. So despite the fact that we had some non-negligible signal in our control, we still were able to demonstrate the specific uptake of our dye to bone using a pre-targeting strategy where the click reaction is happening in vivo. And this, to the best of our knowledge, is one of the first successful uses of pre-targeting for photoacoustic imaging, which has been really exciting for us. So the data that I've shown here today has demonstrated the specific bone binding of our dye using 3 different delivery strategies, including local administration, as well as intravenous active and pre-targeting strategies. We were able to achieve high contrast and spectrally verified signal within the bone in vivo and verified that signal ex vivo as well. So some of the next steps for this probe include evaluating the toxicity in healthy mice, as well as evaluating the ability of the probe to distinguish between healthy and diseased states. So a few of the diseases of interest for a probe like this would encompass any sort of bone cancer, osteoporosis, Paget's disease. Microfractures are something that we're really interested in. Infection-related bone damage, which could be pretty useful when considering the increased prevalence of infections related to medical implants. And finally, one that I think could have a lot of potential for clinical translation is detection of pathological breast calcifications. So with that, I would like to thank you all for your attention, and I would be more than happy to take any questions.
Great job, Rowan. Really interesting work that you're doing. I'm sure a lot of people would be interested in potentially using ultrasound and photoacoustics to look at bone remodeling in a variety of different disease states. Just as a reminder to our audience, if you have any questions for Rowan, please enter them in the Q&A. So we do have a few questions that have come through. Do you think that there's any merit to using the local injection strategy for any other reasons?
Yeah, I think so. So for the purposes of our work here, we've definitely utilized it mainly to show the specificity of our bisphosphonate. But recently, there has been a literature precedent for the use of near-infrared dyes in light-assisted phototherapies of bone. So in theory, you could use something like this, especially the bone targeting nature of our probe, as well as the localized injections, to introduce the dye directly at sites of concern and have your therapy, your light therapy take place. You can see that we were able to get multiple irradiations out of the material, which is promising for therapeutic applications. And as well, it did appear that without the bisphosphonate, we weren't able to keep the dye in place very well. So the fact that we have the bisphosphonate there seems to be crucial in allowing for a compound like this to stay in the area for over the course of a treatment. So I think it could have some merit there.
Great, thank you. Can you comment, do you have any other TCO targeting vectors that you're hoping to use with this dye?
Yep, so we have a bit of work going on in our group right now using antibodies and other small molecules for cancer and infection imaging. So we're only in the preliminary stages of this work, so I couldn't really comment on the success, but given how well it worked with bone, I'm hopeful that we'll have some other avenues there with our TCO antibodies and infection targeted agents.
Great, thank you. Can you comment as to why did you use the 1 hour interval between injections of the TCO-BP and the IR-783?
Yes, so the TCO bisphosphonate was, as I mentioned, previously evaluated within our group. So there's a study that was published where the bisphosphonate compound was radiolabeled with technetium-99m, and a biodistribution of the compound was evaluated. So from that study, they were able to see that the TCO bisphosphonate was fully out of the blood by about 1 hour, and we had decent uptake in the bone at that time as well. So that's why we decided upon the 1 hour, although it does appear that the bisphosphonate stays at bone out to extended time points, such as 24 hours. So we could theoretically change that interval, but we decided on 1 hour since it would be ideal in more of a clinical setting for same day imaging. So yeah, that's why we went with that.
Great, thank you. Can you comment as to any attempts that you made to image any of the organs with the proposed targeting molecule?
Yes, so we did look extensively at the liver and the spleen because for those of you who have worked with these near infrared dyes, such as IR-783, as soon as you introduce any sort of variations in the backbone, like what we've done with our IR-783 tetrazine, we do experience some aggregation tendencies with these compounds. So we definitely were checking out the livers and the spleens to see if the compound was being filtered out there, and they certainly were. We had some pretty green livers, and we're always looking for alternative formulations to kind of help us with the aggregation issues that we've been experiencing. And some of the future work that I do have planned is to introduce some novel hydrophilic dyes that have various functional groups that would hopefully prevent those aggregation tendencies, and then we would maybe be able to have a closer look at and compare the two. We're also working on radio labeling some of these compounds, and hopefully that would give us a bit more insight to full body distribution as opposed to what we were able to do with these studies, which was just to look at a few of the organs at a time.
Great, thank you so much. Well, it doesn't appear that we have any other questions that have come through the Q&A. If you do have questions for Rowan that come up after the webinar today, please feel free to email Visual Sonics and we will happily pass those over to her. So with that, Rowan, thank you so much for the wonderful presentation today. It was great to have you and really fascinating work that you're doing in your group there. As a reminder to all of our participants, if you'd like to connect with us, please reach out to us on our website or any one of our social media channels to learn more about this recording or any of the other information that we have available through Visual Sonics. Thanks so much.